Hello everybody and welcome to the Med-Ace Biostatistics course for medical students. This is Chapter 3 of Section 1. In the last chapter, Chapter 2, we spoke about the research question, types of variables, and types of data. In this chapter, we're going to continue our overview of the basic elements of health research. We're going to talk about the concepts of precision, accuracy, reliability, internal and external validity. Specifically, we're going to discuss the definition of validity or accuracy and reliability or precision. Then we're going to talk about the threats to validity and reliability. Next, we will go over how to measure or assess validity and reliability. After that, we will define internal and external validity. And lastly, we will finish off with an overview of the threats to internal and external validity. We're going to start with a concept which is a bit more abstract, but extremely important in research. We're going to talk about validity and reliability. So what is validity? Validity is defined as the measure of how well the results or data represent the process being studied. It is also referred to as accuracy. Validity decreases as the risk of systematic error or bias increases. Now let's talk about reliability. Reliability is defined as the reproducibility or consistency of the measurements being made. It is also referred to as precision. Reliability decreases as random error or chance error increases. We will talk more about systematic error and random error in just a minute. For now, just understand that validity and reliability are important concepts because researchers always want to produce studies with a high degree of validity and reliability. Let's use this graphic to conceptualize the relationship between reliability and validity. In order to understand this image, we need to assume that the center of the target or the bullseye represents the true effect which the study is trying to measure, and the red dots represent measurements made by the study. In Figure 1, we can see that the measurements or dots are scattered everywhere. Basically, the measurements are all over the place. There's no consistency between the measurements, therefore the measurements have low reliability or precision. Let's contrast this with Figures 4 and 2, in which the measurements are all very similar and consistent. These measurements have high reliability or precision. Now let's talk about validity. Let's look at Figure 2 and 4 one more time. As we said before, both of these images have high precision because the measurements are consistent. However, we can see that in Figure 2, the measurements are all off from the bullseye or the true effect, whereas in Figure 4, they are all in the bullseye. Because the measurements made in Figure 4 are all within the true effect zone, this study has high validity. In other words, this study correctly measured the values that it intended to measure. We can see how a study with high reliability and validity, such as the one in Figure 4, is very effective at correctly measuring the true value every time. This is why researchers pay so much attention to the abstract concept of validity and reliability. Researchers want a study with high reliability and validity so that it measures the true effect every time. Researchers spend a lot of time considering these parameters during the planning phase of the study, and many of the techniques and statistics used in health research are to ensure the highest level of reliability and validity. However, despite all these efforts, many health studies still suffer from poor validity and reliability. So if researchers spend so much time and energy trying to increase validity and reliability, what causes the validity and reliability to be low despite their efforts? The answer is errors in the design and execution of the study. There are usually so many potential sources of error that it is very difficult for researchers to account and control for all of them. Research errors fall into two main categories: systematic error and random error. We're going to spend a lot of time talking about systematic error and random error in the next few chapters. For now, just recognize that random error tends to decrease precision while systematic error tends to decrease validity. Okay, so far we know that researchers strive for studies with high levels of validity and reliability. But how do researchers assess or measure if their study measurements have a high level of reliability and validity? Well, there are a couple of ways. Let's start with reliability or precision. How do researchers assess the level of reliability in their study? They can use statistical tests to measure the level of precision in the measurements of a study. This is done all the time in health researchers, and it is represented by a p-value or a confidence interval. In the simplest of terms, the p-value is a measurement of the probability of seeing a certain measurement or measurements due to chance or random error alone. Random error can be conceptualized as the spread between the measurements. The less random error there is, the less spread there is between the measurements. And the less spread there is between the measurements, the higher the precision. Therefore, random error can be thought as an inverse measure of precision. The higher the random error, the lower the precision, and vice versa. So going back to the p-value, we said the p-value was a measure of the probability of observing a measurement due to random error. Therefore, a p-value can be thought as a measure of random error. This is why researchers always want measurements with low p-values, because it indicates low random error and therefore high precision in the measurement. So how do you increase precision? You can increase precision by increasing the size of the sample. What we have just discussed is not easy to understand. Do not worry, we will talk about this significantly more, especially in section 2. For now, just know that precision is measured using p-values and confidence intervals, which are generated using statistical tests. And also, that the easiest way to improve reliability is to increase the sample size. Okay, now let's talk about validity and how to assess or measure the level of validity or accuracy. In this image, the distance of the measurements from the true value represents the level of accuracy, in which smaller values represent higher accuracy. Unlike precision, researchers cannot use statistics to measure the validity or accuracy of the study. The best they can do is ensure that the study design has no potential sources of bias. In other words, the only way to improve validity or accuracy is to use a study design which minimizes the risk of systematic error or bias. In order to decrease risk of bias, researchers use techniques such as randomization, probability sampling, standardization, blinding, and many more. If a study design accounts for all possible sources of bias, it is said to have a low risk of bias. A study with a low risk of bias is assumed to have high validity. Note that there is no way to know for sure if a study is biased. The best that researchers can do is assume that there is no bias. Okay, now let's apply what we have learned. A group of researchers perform a study to determine the average glucose levels of patients admitted to the ICU. They obtain the following results from a sample of 100 patients. Mean 214, standard deviation 45, and 95% confidence intervals 205 to 223. Another group of researchers in a different hospital performed the same study and obtained similar data to the first researchers. However, their sample size is 1,000. They obtain the following results. Mean 214, standard deviation 45, and 95% confidence intervals 211 and 216. In this example, two groups of researchers perform the same study and obtain extremely similar data. In real life, this is extremely unlikely, but let's pretend it were to happen. The only difference between the two groups is, of course, the location or the hospital and the sample size. The second group used a sample size that was 10 times the size of the sample in the first group. Okay, so now let's answer the question. Which group of researchers have the highest level of precision? Like we said before, we can use statistics to measure the level of precision in the measurements made by the study. In both of these studies, we are given confidence intervals. We can see that the second group of researchers came up with confidence intervals that are narrower than those in the first group. Therefore, the second group of researchers have the highest level of precision. As we will learn in Section 2, the confidence interval reports many kinds of parameters, and one of these parameters is precision, which is represented by the width of the confidence interval. Now I want you to figure out why the second study got significantly narrower confidence intervals if their measurements were basically the same as those in the first study. If you thought because of the larger sample size used by the second group, then you are correct. Remember, as sample size increases, so does precision. Now on to the next question. Which group of researchers have the highest level of accuracy? Okay, so now they're asking us about accuracy. In order to judge the accuracy of the results, we would need to know the true average glucose levels of patients admitted to the ICU. Since we are not provided with this information in the question stem, then there is no way to determine which study had the highest accuracy. Many times in health research, this is the case. Researchers do not know the true value of what they are trying to measure. In these cases, the best thing that can be done is to assess the study design for risk of error or bias. If the study design is found to have a very low risk of error or bias, then the results are assumed to have a high degree of accuracy. Remember, unlike precision, we cannot use statistical tests to assess accuracy. Okay, let's look at another example. A biotechnology company is developing a new point-of-care test for cardiac troponins. In order to determine the performance of the test, a serum sample with a known troponin concentration is used. The test is performed 5 times using the same sample. The following results are obtained: 0.8, 1.1, 0.9, 0.8, 1.2, and 1.0. The troponin concentration of the serum sample was 5.0. Okay, so unlike the previous example, in this example we are given the true value of the process being measured, which in this case is the concentration of troponin. Since we are given the true value, now we can assess the level of accuracy. Let's start with precision. We are given 5 measurements which all seem to be very close to 1. Since these results seem to be reproducible and consistent, we can conclude that the test has high precision. Now let's talk about accuracy. We are told that the true value is 5. This is pretty far from the test results of 1, especially considering that cardiac troponins have reference ranges between 0 and 0.05. Therefore, we can conclude that the test has low accuracy. If the average test results were closer to 5, say ranging from 4.5 to 5.5, then maybe we could argue that the test has good accuracy, but that's not the case. Now let's talk about the different types of validity. When talking about validity, there are generally 2 kinds of validity. Internal validity, which refers to how well the study measured the measures it intended to measure. For example, how close are the blood pressure measurements recorded to the actual blood pressure values of those in the study. And external validity, which refers to how well the study findings can be generalized to the subjects who did not participate in the study, or the target population. For example, how representative are the blood pressure values recorded to those of the entire population? This is an extremely high-yield concept, so let's take a closer look at internal and external validity. In order to conceptualize this, let's imagine a hypothetical study which aims to measure the color of study participants. Let's assume that the study concludes that the study participants are green. If it is somehow determined that the study participants are indeed green, then the study is said to have high internal validity because it correctly measured what it intended to measure, or the color of the study participants. Now let's assume our hypothetical study has very high internal validity. The question remains though, does the study have high external validity? Studies can have high internal validity and low external validity. This is very possible. In order to determine external validity, we can assess the study population or sample to see how similar or representative they are of the target population. In other words, Are the study subjects similar to the target population? If the sample population is similar to the target population, then it is said to be representative of the target population, and it can be concluded that the study likely has high external validity. As we will soon see, the biggest threat to external validity is a type of bias called the selection bias, which occurs when the study sample is not representative of the target population. We will dedicate an entire chapter to bias and study populations, so it is okay if you do not completely understand this topic right now. For now, just recognize that the similarity or representativeness of the study population to the target population are the main factors which determine external validity. Okay, so now let's talk about the threats to internal validity. Like we said before, bias and systematic error are the main factors which drive validity down. However, bias is a very broad term, and many different kinds of bias exist which affect internal validity or external validity preferentially. Therefore, it is important to recognize which type of bias does what. The biggest threat to internal validity is bias that occurs due to poor measurement techniques. This type of bias is called measurement bias. Selection bias, random error, and confounding bias are also threats to internal validity. Selection bias occurs when the study sample is not representative of the target population. Chance or random error occurs due to the individual variation between measurements. Confounding bias occurs due to a third variable. There are many different kinds of bias and study errors. We're going to dedicate a lot of time discussing the different kinds of bias and error later in this section. For now, just understand that any kind of bias or error can significantly decrease the internal validity of a study. The two biggest threats to external validity are the selection bias and poor internal validity. The selection bias occurs when the sample population is not representative of the target population. Like the measurement bias, there are many different kinds of selection bias, and we will talk about them later in this section. However, a very important thing to realize is that you cannot consider external validity without first considering internal validity. If a study has poor internal validity, then it will most certainly have poor external validity. Internal validity precedes external validity. To understand this, let's take another look at our hypothetical study. So now let's imagine that the study has poor internal validity. That is, instead of it rightfully concluding that the study sample is green, it concludes that it is yellow. Given that the study is designed to use a sample which is representative of the target population, then it is very likely that the target population is the same as the sample population. That is, the target population is also green. Therefore, if the study is wrong about the sample population, then it will also be wrong about the target population. In other words, the study conclusion that the study subjects are yellow also does not apply to the target population. So what you need to know is that a study cannot have low internal validity and high external validity because internal validity precedes external validity. Okay, so let's look at another example. A group of researchers is interested in knowing the effect of asbestos on pancreatic cancer risk. They recruit a group of people who have pancreatic cancer and a group without pancreatic cancer, and then perform a questionnaire to determine if the people with pancreatic cancer were exposed to asbestos at a higher rate than those who did not have cancer. At the end of the study, they determined that the participants with pancreatic cancer tended to answer yes to almost every question, while those who did not have cancer almost always answered no. Is this finding a threat to internal validity, external validity, or both? So the type of study design in this example is called a case control study. And if you know something about case control studies, is that they tend to suffer from a kind of bias called the recall bias. The recall bias is a type of measurement bias, and as we said earlier, measurement bias is very bad for internal validity. The recall bias occurs when subjects with the outcome of interest tend to overestimate or overreport the exposure variable, while subjects without the outcome do not report or recall the exposure variable. So the fact that subjects with the outcome variable appear to be over-reporting the exposure variable, while those without the outcome variable appear to be under-reporting the exposure variable, suggests that the study suffers from recall bias. Therefore, since the risk of measurement bias is very high, the study likely has very low internal validity. And remember, if a study has low internal validity, then it also has low external validity. Here we can see a diagram which shows the different kinds of errors and their impact on validity. As you can see, selection bias and measurement bias are all very broad categories, and many different kinds of selection and measurement bias exist. Generally, any kind of study error can impact internal validity. On the other hand, the selection bias and poor internal validity are the biggest threats to external validity. Both internal and external validity can cause a study to make incorrect conclusions. We will take a closer look at this diagram in our chapter on bias and error. Okay, so with that we are done with chapter 3. Here are the take-home points. Validity is the measure of how well the results or data represent the process being studied, and reliability is the measure of the reproducibility or consistency of the measurements made. Internal validity refers to how well the study measured the measures it intended to measure, and external validity refers to how well the study findings can be generalized to the target population. Systematic error or bias and random error or chance are the main threats to validity. Random error can be minimized by increasing the sample size, and systematic error can be minimized by using a study design which accounts for all the possible sources of bias. Lastly, internal validity always precedes external validity. Thank you for watching, and see you in the next chapter.